For some reason, this time it didn't come up. All right. Are there any questions before we begin? Boy, my hair is really a mess. Any questions? No questions? Good. If not, let's go ahead and get started. So today is January 31st, the last day of January, and we're supposed to be starting chapter 5. Did we finish chapter 4? Let me check on that. If anyone knows where we ended, that's fine. Let me see if I have a two.
In my notes, I have we ended at eukaryotic cells. And I think I remember you said we'll get to what we get and then we'll skip the rest of them.
Yeah, we're going to skip
Okay.
eukaryotic cells, but there is a topic I need to talk about. Oh, here it is. Let me see where we got to. So we're a little behind schedule. Why is that not working? I don't know if you guys can see my screen yet, but oh, there it goes. I'm trying to figure out where we got to. All right, so yeah, we had gotten to eukaryotic cells. We are a little behind schedule. You have covered eukaryotic cells in the previous class. And so I frequently do not talk about the different parts of the eukaryotic cell, like the nucleus, the endoplasmic reticulum. Let me see if there's anything I want to talk about. I know there's one topic I want to talk about. I will want to talk briefly about the eukaryotic endomembrane system, and that is that certain parts of the cell are connected, and it has to do with mainly the manufacturing of proteins and then the shipping of proteins around the cell. But it's actually true of other products that the cell makes, like lipids. We'll talk about it with the manufacturing of proteins and but it can happen with other products the cell makes. As far as I know, it's only proteins and lipids, but it might be carbohydrates and I just don't know about it. And that is when proteins are made, they are made by, of course, the ribosomes. And if the ribosomes are attached to the ER, that makes the ER look rough because of the bumps of the ribosome on the ER. With the rough ER making the proteins, they package the proteins up in a vesicle and then send the proteins in the vesicle, meaning the vesicle moves into the cell, to the Golgi apparatus. And it begins on one side of the Golgi apparatus. And then the proteins are modified in the Golgi apparatus. And that could be like an example could be a protein that has methionine as the first amino acid. And this protein doesn't want methionine as its first amino acid. So the methionine could be removed. And there's other things you can do to modify the proteins, such as methylate some amino acids in the protein. Anyways, you modify the proteins. That's the important part. And then the proteins move from this area of the Golgi apparatus to this area of the Golgi apparatus. And when they get to this area of the Golgi apparatus, the proteins are then packaged and secreted from the Golgi apparatus as a vesicle. So the proteins are in a vesicle the second time. The first vesicle budded off of the rough endoplasmic reticulum. I guess I didn't state that. And then the vesicle moves to the Golgi apparatus. And then the protein moves through the Golgi apparatus. And on the far side of the Golgi apparatus, meaning there's a side of the Golgi apparatus that's got smaller blocks of the Golgi, and the other side is where the bigger blocks are. This is the side that sends out the proteins in vesicles. And then the protein moves to where it's supposed to be in the cell. Now, for some proteins, they will be taken to the cell membrane, and then the vesicle of the membrane of the vesicle will fuse to the membrane of the cell membrane. And the two are both made out of phospholipids. That's why they can fuse together. And that membrane of the vesicle will just become part of the membrane of the cell membrane. And that will then open up the proteins for export out of the cell. Other places, the protein can be moved to another place in the cell, the place where the protein is supposed to be, and then the protein will be used in that region of the cell. For example, let's say an enzyme in the nucleus, the vesicle will move to the nucleus and then fuse with the nuclear membrane and then release the protein, which are enzymes that are going to work in the nucleus. And then the enzyme will be in the nucleus and those proteins will function in the nucleus. The point is the vesicles are moving proteins around from one location to another. And in the endomembrane system, we have vesicles twice, once coming from the rough ER to the Golgi, and then from the Golgi to wherever the protein is going to end up. If the protein is kept for storage, the vesicle will move to some location in the cell and then just store the protein in the membrane that used to be a vesicle. However, since the vesicle is no longer moving, we don't call it a vesicle anymore. Once it's changed from the function of moving to the function of storing, we call it a vacuole. And so if a vesicle moves to a location in the cell, while it's moving, we call it a vesicle. But once it stops moving and it's just storing the protein for, I don't know, later use, then we call it a vacuole. Any question about any of that? All right, that's the endomembrane system. And obviously that's working in a eukaryote cell because we have endoplasmic reticulum and Golgi, as well as vesicles. You don't have any of that in a prokaryotic cell. I should briefly mention that the mitochondria and the chloroplast are different membrane-bound organelles for two reasons. They are a membrane-bound organelle that has two membranes. I'll just talk about it with the mitochondria. The chloroplast is similar, it's just much more complex. Come on, there we go. And that is the mitochondria has an outer membrane, so does the chloroplast. And then it has an inner membrane, which is highly folded on itself.
Professor, sorry to interrupt you, but I don't think we can see your screen.
Oh,
If ever you're referring to something.
I've been referring to something for a long time, so you should have mentioned it earlier.
I don't mean to disrespect, but yeah, I
Yeah,
don't mean to
no,
cut you
that's
off.
fine. No,
Thank you.
that's fine. You should have interrupted. Let's go share the screen. All right. Now you can hopefully see the screen.
Yes.
Let me briefly go back and show you the picture of the endomembrane system. It's not that complex, but I was showing that this is the rough ER where vesicles bite off of it, bringing the proteins to the Golgi apparatus, and they join on this side of the Golgi apparatus. The proteins are modified by the Golgi, and then they vesicles take the proteins out of the Golgi apparatus and take the protein to where it ends up in the cell. And that's the picture I was trying to show you there. All right, so let's go to the mitochondria. It has two membranes, and that is the outer membrane, and then an inner membrane, which is highly folded to increase the surface of the, to increase the surface of the membrane. And the reason for that is in the mitochondria, that's where the ATP is made. And apparently the mitochondria needs more space for making ATP. That's not terribly important. That's one of the differences between the mitochondria and the chloroplast from other membrane-bound organelles. The other difference, and I guess there are a couple, but the other main difference is these membrane-bound organelles, the mitochondria and the chloroplast, have a DNA molecule inside them. Okay. All right. Those are the two main differences, and I just was going to point that out. And now let's talk about the endosymbiotic theory, explaining how mitochondria and chloroplasts came to be in eukaryotic cells. How did these organelles originate? And it also explains why these organelles, membrane-bound organelles, have two membranes, an inner and an outer one, as well as why they have DNA molecule inside them. Okay. I'm going to open up Paint to give my story of the not the endomembrane system. Endosymbiotic theory. Just a minute, let me close that down. And no, I don't want that. What do I want? Oh, here it is. Paint. A long time ago, when eukaryotic cells weren't quite around yet, there was a proto-eukaryotic cell which was very large, and it engulfed a smaller cell, which was a bacteria. I'm going to sort of make it like a short rod. I didn't draw that very well, but that was brought into the cell. And then for whatever reason, the proto-eukaryote did not eat the bacteria cell that it engulfed. And then it, that's actually a little bit of a rod. And it had two membranes around it. One would be the vesicle membrane when that cell was brought into the proto-eukaryotic cell. And then it had, of course, the cell membrane of the bacteria. And for whatever reason, the proto-eukaryotic cell did not eat the cell right away. And so this vesicle was inside the cytoplasm of the prokaryotic cell. And inside the cytoplasm, we had glucose. I don't know if that was a good thing to do or not, but the proto-eukaryotic cell, let's do it over here. The proto-eukaryotic cell, I'm going to go abbreviate, could not perform aerobic respiration, and all it could do was something like fermentation, meaning it could make one molecule of glucose and make two molecules of ATP by fermentation. The bacteria, oh, I got it already, sorry. The bacteria, on the other hand, could perform aerobic respiration and could take one molecule of glucose and make something like around 38 molecules of ATP. I should blow this up so you can see it better, but sort of stuck with this now. Just blow it up here. Oh, that's maybe too big. Oh, I see what it did. It didn't go in increments. It went from 100 to 200. Let's go one more.
You can select it and make the font bigger.
I can make the font bigger, but I've already made it, so I didn't want to do that again. Gosh, let me scroll over and scroll down. That's all I wanted. There we go, kind of. Anyway, so the glucose from the cytoplasm of the proto-eukaryote cell went into the vesicle, and then the bacteria used the glucose and then made 38 ATP. And probably because life was young and early, there was a lot of mixing, like the glucose moved from the proto-eukaryote to the bacteria. And when the bacteria made the ATP, it gave some of that ATP to the proto-eukaryote, just putting it in the cytoplasm or secreting it, and then it diffused into the cytoplasm of the proto-eukaryote. And we don't know how many ATP it made, but the proto-eukaryote said, geez, I lost one glucose, but I gained a number of ATP. And let's just make it something like half of 38, whatever that number is. Let's see, 40. So it's about 19. And we don't know exactly what it is, but it was a large number, much larger than the 2 ATP that the proto-eukaryote could make itself out of 1 molecule of glucose. Okay. And so the proto-eukaryote said, geez, I should keep this bacteria around because it's making me ATP, a lot more ATP than I can make. And where's my scroller bar? I don't see it now. Oh, I've got to scroll down here probably to get it. Nope. For some reason, my mouse is not finding the scroller. Oh, it's because I'm in that box. Okay. And so the proto-eukaryote kept the bacteria around. The proto-eukaryote gave a home to the bacteria, supplying the bacteria with glucose. And then the bacteria would take the glucose and make ATP and release at least some of the ATP for the proto-eukaryote. And then the two started living in symbiosis. That's where the two live together and live in harmony, meaning the proto-eukaryote made a home for the bacteria and the bacteria made glucose for the proto-eukaryote. The two have lived in symbiosis now for so long, we do not find this species of bacteria outside of the proto-eukaryote cell. And where do we find the proto-eukaryote? Well, now it is a eukaryote, and the bacteria is no longer called the bacteria, it is called the mitochondria. And that's the theory of the endosymbiotic theory. What is the name of that? Endosymbiotic theory. The two organelles were separate species. They lived together in symbiosis for a long time. And now we only find the bacteria inside what used to be the proto-eukaryote cell, but the proto-eukaryote cell now has a mitochondria, and we call it a eukaryotic cell. Any question about any of that? That's the theory. Now, this theory actually happened more than once, and that is in animal cells, they got a bacteria in them, which became the mitochondria. In plant cells, they actually got a bacteria in them which became the mitochondria, and then they got a photosynthetic bacteria in them which became the chloroplast. And the chloroplast is similar. It has two membranes around it, and it has a molecule of DNA inside of it, just like the mitochondria. All right, so what evidence do we have to support the endosymbiotic theory? Well, first, we have some eukaryotic cells, they're always protists, which can engulf another cell like this. What is that? Paramecium has engulfed a photosynthetic bacteria, or sometimes it's an algae, and then the two can live in harmony. The protist, the paramecium, provides a home for the photosynthetic cell, which in this case is a bacterium, and then can supply the bacterium with nutrients and food. And then the bacteria, because it's photosynthetic, can engage in photosynthesis, making glucose, and then share that glucose with the protist, the paramecium. Okay, and there's actually a few species of amoeba which lack a mitochondria, and these species of amoeba will engulf a bacteria and then use these bacteria to serve as a mitochondria. So there's actually certain amoebas, I don't know how many species there are, but it's one family of amoeba that do not have mitochondria, and they engulf a bacteria, and then the bacteria engage in aerobic respiration to make ATP for the amoeba. And the amoeba gives the bacteria a home as well as nutrients. And then the bacteria makes ATP for the amoeba. So we actually see, I don't know what we're saying happen. We actually see it today. And then some other evidence for the fact that the theory that the mitochondria and the chloroplast were once free living cells, which then became incorporated into a proto-eukaryote, which evolved into the eukaryote, is shown here. First, I mentioned that the mitochondria and the chloroplast have two membranes. You would expect two membranes if a cell, like a bacteria, were to come into a larger cell like the proto-eukaryote or the eukaryote, because it should be surrounded by the cell membrane and then the vesicular membrane, the membrane of the vesicle. Okay. When we look at the growth of the chloroplast and the mitochondria, they divide by binary fission, the same way that prokaryotic cells or bacteria cells grow by. On the side, eukaryotic cells do not grow by binary fission, they grow by mitosis. When we look at the ribosomes that are made by the mitochondria and the chloroplast, they are 70S in size. And there is a gene on the DNA found in the mitochondria and the chloroplast, which encodes this 70S ribosomes. And this is the same size as the ribosomes of a prokaryotic cell. Remember, the eukaryotic cell has a larger ribosome. When we look at the first amino acid that is made in protein synthesis in eukaryotic cells, it's methionine. I haven't ever mentioned it, but in prokaryotic cells, it's not methionine. It is a related amino acid, and it's formylmethionine. This is the first amino acid that is made when a protein is being made from the ribosome, meaning AUG. In eukaryotes, codes for methionine, and in prokaryotes, it codes for formylmethionine. In the mitochondria and the chloroplast, it also codes for formylmethionine. When we look at the presence of histones, you find them in a eukaryotic cell's DNA. They're missing in bacteria, the histone proteins, and they're missing in the DNA of mitochondria and chloroplasts. When we look at the type of DNA, eukaryotes have linear chromosomes. Bacteria generally have one circular chromosome, and the DNA of mitochondria and chloroplasts are one circular chromosome. And then furthermore, when we sequence the DNA found in the mitochondria and the chloroplasts, the sequence of the DNA does not fall in the domain eukarya. The sequence of the DNA of the mitochondria and the chloroplast falls in the domain of bacteria. Okay. All right. Any other questions about evidence supporting the, oops, sorry, didn't mean to do that. Let's get rid of that. The endosymbiotic theory. All right, there's a few other evidence I'm not going to go into, and that is the DNA of eukaryotes have introns and exons in a gene, and prokaryotes, or at least bacteria, do not, and the mitochondria and the chloroplast do not. But the biggest is when you sequence the DNA of the chloroplast and the mitochondria, they fall within the domain bacteria. All right, any questions about the endosymbiotic theory? All right, that's it for chapter, I don't know where I changed that, so I'm not going to do that. That's it for chapter 3, I think. 4. Let's move on to chapter 5, microbial metabolism. We're not too terribly far behind, just a little bit. At the end of the lesson or in the lab, somebody remind me to make chapter 4, the notes, the PowerPoint available to you. There wasn't a preliminary slide, so I knew something was wrong. All right, microbial metabolism. This slide is showing you the major goals and a rough outline. Obviously know the terms of microbial metabolism, understand what feedback inhibition is, and we're going to talk about respiration, especially aerobic respiration, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Any questions about what we're going to cover? All right. When we're talking about metabolism, it's important to note that metabolism is all of the chemical reactions that occur within an organism. And for microbes, since an organism is a single cell, metabolism is all of the chemical reactions inside a cell. You can break metabolism into 2 parts, catabolism and anabolism. Catabolism is a decomposition reaction where a larger molecule, such as starch, is broken down into a simple, smaller molecule, such as glucose. Energy is given off by this reaction, and we can make ATP from some catabolic reactions. Now, this slide is showing you that catabolism happens with the making of ATP you should realize that animal cells are really lousy at making ATP. We only have two chemical reactions that make ATP, and that is aerobic respiration for animal cells, and then fermentation. In all other catabolic reactions, which are by and large the vast majority of them, you do not make ATP in animal cells. Any question about that? So the slide is a little oversimplified. And like I said, there's only two chemical reactions in animal cells that make ATP. All of the other catabolic reactions like, I don't know, removing an amino acid from a protein, actually even moving one glucose from a starch would not, an animal cell could not make ATP out of that. Okay, it's actually only the breaking down of glucose, or breaking down of starch into glucose and then breaking down the glucose that makes ATP, or the fermentation. All other catabolic reactions do not make ATP. Now, when you're transferring energy from one source to another, such as from starch to glucose, some of that energy will be converted into heat. And heat is a form of energy. It's just not terribly useful to cells because cells cannot use heat for their energy needs. And that's true for humans too. But we can use heat, at least warm-blooded animals, actually even cold-blooded animals, can use heat to help warm up the body if you need more heat. Like when you're shivering, you're burning molecules and generating heat to help warm up the body. But usually that heat is just waste and it's lost. The cell can't use the energy and heat for anything else. So the catabolic reactions release energy. They always release energy, and that's because the larger complex molecules store more energy than the smaller, simpler molecules. Anabolism is the reverse of catabolism. It is a biosynthesis reaction where you take a smaller molecule, such as glucose, and you take many of them, and then you build a larger molecule, such as starch. A biosynthesis reaction. This reaction takes ATP to give the energy it needs, meaning we're starting with a molecule or several molecules that have less energy in them than the large complex molecule. So this reaction requires energy for the reaction to occur. So this is an energy-using reaction, anabolism. Catabolism is an energy-releasing reaction. Now, once again, when you convert one form of energy, such as the energy in glucose and the energy in ATP, into the energy of starch, some of that energy is lost and is lost as heat. So whenever you change one form of energy to another, you always lose some of that energy as heat. And if you don't know, that's the second law of thermodynamics. I'm not going to quiz you on that, but you should know that from a previous course. Any question about anabolism and catabolism? All right. Anabolism requires energy for the reaction to proceed. Usually that energy comes from breaking ATP, which releases the energy for the anabolic reaction. And then the ATP is converted into ADP and inorganic phosphate. When we're talking about metabolism, we can link different metabolic reactions together to get a metabolic pathway. Let me blow this up a little to make it more easy to see. So we can have molecule A, which is converted by an enzyme into molecule B. Molecule B can be converted by another enzyme, enzyme 2, into molecule C. Molecule C can be converted by an enzyme into molecule D. Molecule D can be converted by an enzyme to molecule E. Molecule E can be converted by another enzyme into molecule F. And then you string all these chemical reactions together, we have a metabolic pathway, converting molecule A eventually into molecule F. Molecule F is the last molecule made, so we call it the end product. Molecule A is the starting substrate, which is also called the reactant, the first molecule you work with. All of the other molecules are intermediates, and you can call them intermediate products because molecule B is the product of the first reaction, but then molecule B is the starting substrate of the second reaction. Okay. All right. Metabolic pathways are determined by enzymes, meaning in a cell, we can have this metabolic pathway work because we have the enzymes in the cell to make the products of the metabolic pathway. And then enzymes are encoded by genes. So ultimately our genes are responsible for our metabolic pathways. Any question about any of that? All right. Here is a slide showing you all the metabolism that is occurring in a cell, and we'll discuss each of these chemical reactions. Any questions? Only kidding. Just telling you there's a huge amount of metabolism going on in a cell. Chemical reactions occur via the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, or molecules collide with each other. The activation energy is the energy needed to disrupt the electronic configurations of the molecules that collide with each other to convert it into the products. So the activation energy is the energy needed to disrupt essentially the chemical bonds of the starting substrate and then convert it into the product. The reaction rate is the frequency of collisions having enough energy to bring about a reaction. So if more collisions happen, you can increase meaning you increase the number of collisions, you will increase the reaction rate because there will be more collisions, more molecules will be converted into the product. You can also increase the abundance of enzymes. The more enzymes you have, the faster the enzymatic reaction will occur up to a point. We'll talk about that, what point it is. And then you can also increase the temperature. That makes the molecules move around more rapidly, meaning the molecules that are moving and colliding with each other will have more energy. And that's just what the temperature is, a measure of the movement of the molecules. And the faster they move about, the higher the temperature. And so if you elevate the temperature, you also increase the reaction rate. A similar matter happens with pressure. If you increase the pressure, you make the reaction more likely to happen. And how do I explain that? It's because you're putting pressure on the molecules, and so they're more likely to collide and collide with more force because they have more pressure between the molecules. And if you don't understand the pressure part, don't worry about it. Just try and understand increasing the temperature increases the reaction rate. Any question about any of that? All right. So this chart is showing you the activation energy of a chemical reaction. We start with substrates, which have a certain level of energy in the molecule. And then we end with the products, which have another level of energy in the molecules. In this case, we have the products having less energy than the substrate. And so this could be something like burning of glucose, which has a lot of energy in the molecule, forming water and carbon dioxide, which they do have some energy in those molecules, but it's much less than the energy in the glucose. In this case, energy is given off by the reaction, and that would be the energy from here to there, which isn't shown on here. The point shown here is that for the substrate to be converted into the product, we have to go over an energy hump. And there are two possible energy humps. One is the reaction without an enzyme, which requires the molecules to gain much more energy before the reaction will proceed. And that is called the activation energy without an enzyme. The other is the reaction with an enzyme, and it too has an activation energy with an enzyme, but it is much less than the activation energy without an enzyme. And it has an energy hump, meaning you do have to add energy to it, but it's much less before the reaction will occur and then we get the products. An example of this would be to burn glucose. You could take a cup of glucose and take a blowtorch to it, and that will add energy to it, and that would be the activation energy without an enzyme. And as you know, a blowtorch is really hot. You're adding an extreme amount of energy to the glucose, but that will burn the glucose and convert it into CO2 and water. There's another way that this happens, and it's happening in your room all the time, where glucose is being converted into CO2 and water. And where is that happening in your room right now? Come on, somebody's got to know, somebody's got to guess. I'm not going to tell you. So where is that happening in your room? Is it happening in your Bunsen burner if you have a Bunsen burner?
We're breathing and making oxygen and CO2.
Inside your body, in your cells, we are converting glucose into CO2 and water. So that's happening all the time in your room. And is that temperature, the activation energy with an enzyme, anywhere near the temperature of a blowtorch? That's a yes or no.
No.
Yeah, it's a no. It's happening at 37 degrees, which is 98.6 degrees, if I remember correct, Fahrenheit. And that's much lower than the temperature of a blowtorch. And that's showing you that enzymes really help perform a chemical reaction. You don't need to convert this up very much. You only need to bring it to 37 degrees to get the activation energy with an enzyme. You don't need to take it, whatever the temperature of a blowtorch is. It's something like 1,000 degrees or close to that. I don't know what it is, okay? Much less. And that's showing you how helpful enzymes are in making a chemical reaction occur. All right, Enzymes are biological catalysts. They speed up a biochemical reaction. Many times, if you really want to know, it's up to 10 to the 8th times, whatever that number is, 100 million to 10 to the 9th times. I think that's 10 billion times. And you don't need to know these numbers. It's just telling you that enzymes and biological catalysts can really speed up a chemical reaction. Meaning I don't know if you can see this, have a cup here, put glucose in it, and eventually all of the glucose will be converted into CO2 and water. But that's not going to be anytime soon, is it? Meaning if I were to look at this cup and measure it out tomorrow, I'd still have a cup of glucose. And if I leave it for 2 days, I'd still have a cup of glucose. If I were to leave it in here for a year, it would still be a cup of glucose. It would take a long time for that glucose to be converted into CO2 and water. But if you know what is that particle that comes from space? Electromagnetic particle. I'm forgetting the name of it now. That has a lot of energy. It could hit the glucose and then convert the glucose into CO2 and water. But that would only affect one molecule of glucose. And as you know, in your computer, that molecule can hit your computer and then create an error in the, what do you call that? The hard drive of the computer. And our computers last for years, so that molecule does hit our computers, but there aren't that many of them because our computers will last for years, hopefully. And they will, with time, accumulate errors because that particle, what the heck is the name of that particle? Let me do a search online. I don't remember. Oh, I almost had it. It's not a radon particle. Cosmic particle, maybe? Well, not really neutrinos. That's not what I'm looking for. Cosmic rays. That's the particle I was trying to say. When they hit your molecule of glucose, they can convert it into CO2 and water. I don't know if I should close that down or bring it back to yeah, I can close that down. All right, any questions about any of that? Enzymes are specific for a chemical reaction, meaning usually one enzyme will bind to one specific reactant, one specific substrate, and only that substrate, and then bind to it and convert that substrate into a product. For those of you who work with drugs in a clinical setting, you know that that's not entirely true. Sometimes we can have a drug that'll mimic an enzyme, and then we can keep this chemical reaction from happening by taking this drug. And that's because the drug is competing with the enzyme for the substrates binding to it. But for our purposes, we'll just make it simple and say that generally an enzyme has a specific shape that allows it to bind to one specific substrate, and it doesn't bind to other molecules, other substrates. And then it can proceed to form the chemical reaction. And this is due to the enzyme specific three-dimensional shape, where it has a specific region that binds to the substrate. And I'll talk about that specific region in a minute. Furthermore, the enzyme actually binds to the substrate and then forms a enzyme substrate complex. It's the enzyme substrate complex that increases the probability of a reaction occurring by putting stress on the existing chemical bonds of the substrate and then making it more likely that the bonds will break and then form the product. Assuming that we're breaking chemical bonds. You may take two products together and then put stress on them and form a bond, and we're not going to talk about that. Anyways, that is why the enzymes lowers the activation energy. It's because it's actually the enzyme is forming an enzyme substrate complex with the substrate. Any question about any of that? All right. You should know that enzymes commonly end with ASE or ACE. You don't need to know the examples, but we'll talk about, we'll talk about ligase, and we've already talked about, I think, catalase. And you may have run into an enzyme in biology 160. What the heck was that? Catocalase? Enzymes frequently end in ACE, but not all enzymes do, especially the enzymes that were named before the ACE convention was started. Like if you happen to know, renin is an enzyme that curdles milk in your stomach, and renin is an enzyme, but it does not end in ACE. Any question about any of that? All right. You do not need to know the names of the components of an enzyme, but some enzymes need a helper before the enzyme will work. So some enzymes bind with a coenzyme, and they only work in the presence of the coenzyme. We'll talk about enzymes in a minute. A coenzyme is a small biological molecule like NADH is a coenzyme. And some enzymes bind with a cofactor before the enzyme can work. A cofactor is usually a chemical element like iron, zinc, magnesium, copper. So it's a non-protein, well, it's an element, non-protein element. A coenzyme, on the other hand, is a biological molecule. Some important coenzymes include NAD+, which is nicotinamide adenine dinucleotide. Another one we'll talk about, we're going to talk about NAD+ and FAD. These are important coenzymes, FAD, flavin, adenine dinucleotide. These two coenzymes come into play in aerobic respiration, so we will talk about them. And you should know these two as important coenzymes. The enzymes which perform in aerobic respiration of the making of ATP have NAD+ and FAD as important coenzymes. There are some other coenzymes, you don't need to know them. NADP+ you can find in green plants. You don't need to know that one. FMN, you don't need to know that one. Coenzyme A, I don't require you to know that it's a coenzyme. We actually will briefly talk about coenzyme A when we're talking about aerobic respiration, but you only need to know the first two. All right, this slide is showing you that enzymes bind with a substrate to form an enzyme substrate complex, which is shown here or there. And then that puts stress on the bonds of the substrate, which will help the substrate convert into the product, where that bond is then broken in this case. Once the product is made, it falls off of the enzyme, and then the enzyme is then free to come again and bind to the substrate, and then proceed with the chemical reaction again. And that's why enzymes are so good at performing chemical reactions, because they'll just be reused hundreds or thousands, or maybe even hundreds of thousands of times. Okay, any question about any of that? All right, this first slide is trying to show you the lock and key model of how an enzyme works. And that is like a key that fits one lock. The key is specific to that lock. And that's the way an enzyme is like a key fitting one substrate. And that's the lock and the key model, that an enzyme acts like a lock and a key, converting the substrate into the product. Any question about the lock and the key model? All right. This is the hand and the glove model of an enzyme. And the hand and the glove model states that an enzyme works like the hand going into the glove, where the glove changes its shape to fit to the contours of the hand. And if you're looking at this, you can see here's our substrate, and there's the enzyme. The enzyme is changing its shape to fit to the contours of the substrate. And this is the hand and the glove model. What else do you notice about this picture here? There's two things if you look really, really closely. I can't blow it up any further. Nobody's going to state anything? What about the size of the enzyme to the substrate? Anyone notice anything curious about that? Come on, people, don't be shy. The enzyme,
Enzyme's a lot bigger.
yeah, the enzyme is much bigger than the substrate. And that's the way biological enzymes are. Another thing you can do from this, and I haven't stated it, but it's shown up here, is the enzyme actually has an active site which physically binds to the substrate, a portion of the enzyme that fits to the substrate, and that would be shown right here. The other thing, if you look right here and right there, and that is not only is the enzyme changing its shape to bind to the substrate, but the substrate is a little flatter. It's changing its shape to bind to the enzyme. And I like to call this the shapeshifter's hand in the glove model, where yes, the glove is changing its shape to fit the hand, but the shapeshifter's hand is changing its shape to fit the glove. And that is what really happens. The substrate is also changing its shape to fit the glove or the enzyme, and the enzyme is clearly changing its shape to fit the substrate. Any question about any of that? All right, enzymes do not work in a vacuum. Enzymes, well, they work best when they're at the correct temperature, the correct pH, the correct salinity, when heavy metals are not present, when alcohol is not present, and UV radiation is not present. When the temperature is not correct, the pH is not correct, the salinity is not correct, when heavy metals are present, or alcohol is present, or UV radiation is present, the enzyme will not work as well if it works at all. Okay? And with temperature and pH and salinity, when you're close to the correct temperature, the enzyme will work, just not as well as at the correct temperature. And the further you get away from the correct temperature, the less the enzyme will work, until you get to a certain temperature that's so far away, the enzyme will no longer work. And that will happen if you freeze the enzyme, it clearly won't work. Or when you heat the enzyme, it won't work. And it depends on the enzyme what its minimum and maximum temperature is. But most of our enzymes will not work when they're frozen, because our cells aren't going to be metabolically active when they're frozen. But also when you heat them up, they will not work as well, and if at all. And certainly if you were to boil our enzymes, they would not work at all. Okay? When the protein making the enzyme changes its shape enough such that it's not working at its optimal level, then we say the protein has started to denature. And when it's totally denatured, we say the protein has denatured. Okay? You can get the protein to denature at the wrong temperature. Like when we cook an egg, the albumin in the egg whites will be denatured, take on another folding pattern, and that will be the white egg whites that we eat when we cook eggs. And if you were to take the wrong pH, like put the egg white in very acidic acid, just lemon juice, not that acidic, and the whites of the egg whites, would start denaturing, and they too would take on a white appearance, meaning that the proteins have been denatured. And that's true for salinity, the presence of heavy metal, alcohol, and then UV radiation. Another factor that can influence enzyme activity is the substrate concentration. Up to a point, when you increase the substrate, you will increase the enzymatic activity. If various poisons are present, you will decrease the enzyme activity. And if each enzyme is affected by the poison, you will have no enzymatic activity, such as when you put cyanide in the environment. Obviously, cyanide would kill cells, and that would stop the enzyme from working. Any question about any of that? If not, we're going to talk about inhibition. How do you stop chemical reactions? And you can inhibit the chemical reaction. There's two ways to inhibit it. You can have competitive inhibition, and then you can have non-competitive inhibition, which is usually called allosteric inhibition. And I'll use the term allosteric inhibition. And then you can also have feedback inhibition, which is a special case of inhibition. All right, I mentioned that when an enzyme is denatured, it will start losing its function, and the more it's denatured, it will be totally losing its function. Like here's an active functional protein, and it's folded up, taking on a three-dimensional pattern. You put it in the wrong environment, the protein will unfold and become a denatured protein. And this one is totally unfolded. It will not work at all. And you can denature a protein simply by putting it at the incorrect temperature, the incorrect pH, the incorrect salinity, or a slew of other factors. When we're talking about the enzyme's activity, this enzyme works best around pH 37, so that could be one of our enzymes. And as you move away from that, the enzyme still has activity, but it's not the optimal amount of activity at pH 37. And then when you move way away, the enzyme will not have any activity. When the enzyme's at 10 degrees, less than 10 degrees Celsius, 0 is when water freezes. And then 50 degrees Celsius, the enzyme will not have any activity. The same with pH. This pH is optimal around pH 5. So this might be a skin enzyme. And then when you move away from that, the enzyme will have less activity. And you move further away, the enzyme will have less activity. You move into extreme pH 10 or pH 0, the enzyme will not have any activity. And we can say the enzyme is totally denatured. Any questions about any of that? All right. Another factor that can influence the enzyme activity is the substrate concentration. If you increase the substrate concentration, you will increase the enzymatic activity until a certain point, meaning the substrate concentration, you increase it, you increase the enzyme activity very greatly, but then it starts to slow. And then when we get to here, the substrate concentration, when it's increased, will not increase the enzymatic activity at all. Why do you think that might be? What could explain that? It's actually very simple if you think about it. Okay, nobody's taking a guess. Well, let's say we have 10 enzymes in the test tube, and we have 5 of those enzymes bound to substrate. Well, if we increase the substrate concentration, we will increase the enzymatic activity because now more than 5 enzymes will bind to the substrate. However, once all of the enzymes are bound to the substrate, then if we increase the substrate concentration further, you cannot speed up the enzymatic reaction any further because all of the enzymes are already bound to the substrate. Does that make sense? Think about it if it doesn't.
So then you're saying that there's always a limit?
Well, there's a limit when you increase the substrate concentration. There will always be a limit. Once you increase the substrate concentration so that all of the enzymes are bound to a substrate, you cannot speed up the chemical reaction any further once you increase by increasing the substrate concentration.
Okay, thank you.
Okay. And that's also true with the amount of the enzyme. You could increase the enzyme, leaving the substrate at the same concentration, and that would speed up the enzyme to a point, the enzymatic activity to a point. But once again, you'll plateau off, and that plateau off will be when all of the enzymes are bound to the substrate, or all of the substrates are bound to the enzyme. You can't increase the enzymatic activity any further by increasing the amount of enzyme because all of the substrate are already bound. You can't bind anymore. All right, let's talk about competitive inhibition. Competitive inhibition is when there's an inhibitor that competes with the substrate for the enzyme. So in this case, we have a substrate and it's binding to the enzyme at the active site. We can have competitive inhibitor by adding the inhibitor, and this inhibitor will bind to the active site and it will compete with the substrate for that active site. So if you increase the inhibitor, like let's say we're wanting to shut off this enzyme and we're doing it with a drug, we could simply increase the competitive inhibitor and then it will be more likely to bind to the enzyme, shutting down the chemical reaction. Okay? Any questions about that? A non-competitive inhibitor is similar. It's just the non-competitive inhibitor binds to the enzyme, but a site other than the active site. And when it binds to the enzyme, it changes the folding pattern of the enzyme, and now the active site can no longer bind to its substrate. And that's the way a competitive inhibitor works. It works by changing the folding pattern of the enzyme, and then the enzyme can no longer bind to the substrate. Any question about any of that? All right. There are different factors that can influence the amount of enzyme activity, and one is feedback inhibition. Feedback inhibition is a type of allosteric inhibition. So with this metabolic reaction, we have a substrate which is converted to enzyme by enzyme 1 into intermediate A, which is converted by enzyme 2 into intermediate B, which is converted by enzyme 3 into the end product. Okay? Well, what happens if we don't want to make any more end product, but we still have our enzymes around? What you can do is feedback inhibition, where the end product itself can bind to the first enzyme and then change the shape or the folding pattern of that enzyme. So the enzyme will become inactive and it will no longer bind to the substrate. The reason it no longer binds to the substrate is that the active site has changed its shape when the protein binding was changed by right here, I guess, by the allosteric inhibitor, in this case, the feedback inhibitor. So the enzyme no longer binds to the substrate. That means no intermediate A will be made. If no intermediate A will be made, then no intermediate B will be made. If no intermediate B is made, then we'll make no end product, because intermediate B is turned into the end product. And that's the way feedback inhibition works. You should realize that feedback inhibition is a common way that cells regulate their contents. The reason why they do it this way is, first, you don't remove enzyme 1, you just make it inactive. So later it can be activated and then work again. Another reason why you want to hit enzyme 1 is because it's the first enzyme that works. If you were to hit a later enzyme, then you will make at least intermediate A, and that takes energy for the cell to convert the substrate into intermediate A. So you don't want to stop enzyme 2, you want to stop enzyme 1 because it's more energy efficient. And then it's also energy efficient in that the end product is acting as an inhibitor. When you have enough end product in the cell, you want to stop making more end product because you don't need any more. And you can use feedback inhibition where this end product will itself directly shut down enzyme 1 that would eventually make the end product. Any question about any of that? So this feedback inhibition is common in cells. Oh, and the other reason it's common in cells is if you don't have enough end product around, this end product binding to this enzyme here will every once in a while fall off simply by stoichiomic reactions, meaning the binding is not permanent and it will fall off. If there's lots of end product around, one of the end products will bind to the enzyme and keep it shut down. But if the cell needs more end product, there won't be much end product around, and so there will not be an end product binding to the enzyme 1. And then enzyme 1 will be active, and then we'll start making the end product again. And those are the two reasons why feedback inhibition are commonly seen in cells. Come on. All right. I want to talk a little bit about ribozymes. I mentioned earlier that proteins make most enzymes. And if we have an enzyme, it's most likely a protein enzyme. However, not all enzymes are protein enzymes. There are a few enzymes which are made of RNA. They do not have protein in them. And they have a special name for these non-protein enzymes. They call them ribozymes. They are an enzyme. For example, a ribozyme, which is an RNA enzyme, can cut and splice RNA, as well as other molecules. And so it's acting like an enzyme. The only enzyme we'll ever talk about in this class is that is an RNA enzyme, is on the ribosome. When the ribosome is making the protein, it transfers the earlier amino acid to the latest amino acid that's been brought in. And so it cuts the amino acids off of the first group making the protein and then transfers those amino acids to the next amino acid that's put down to make the protein. Anyways, the point is that portion of the ribosome is an RNA molecule and it has enzymatic activity, cutting the amino acids off of the, actually it's a T cell, and then binding those amino acids to the protein in the, where would that happen? The Golgi apparatus? Yeah, the Golgi apparatus. So ribozymes do function as enzymes. They even have an active site that will bind to the substrate. And then the ribozymes are not used up or modified in a chemical reaction. So they are an enzyme and they work the same as protein enzymes, but they're not made of protein. And our bodies only have a few of these RNA enzymes. Any questions about any of that? If not, I think I'll stop here and I'll see you at 6:30 for the lab.